So good morning students. Today we will start with the third day series on the chapter respiration in plants, which is an important chapter for class 11th CBSE, SSC, ICC, as well as for NEET examination or MHCET. So in the two previous sessions, we discussed glycolysis and fermentation in the first video. In second video, that is in the second video, we discussed link reaction and Krebs cycle. Today in this video we will discuss ETS, that is electron transport system. ETS means it's a transport system which we are using in photosynthesis. You may be familiar with this system. What is the basic necessity for this? We have to create proton gradient. We have to create proton gradient. Proton gradient means we have to increase proton concentration we have to increase the proton concentration in the intermembrane space. We have to increase. Yes. We have to increase the concentration of protons in the intermembrane space of the mitochondria. Yes. ETS is taking place in the mitochondria. Simple. Where in the inner membrane of the mitochondria. Two questions for you all. If you are watching my videos or any other videos, so you have to answer this question: where does glycolysis takes place and where does Krebs cycle takes place? Comment your answer in the pause the video and comment your answer. And now I will tell you the answer. Pause, think for a while, revise it. So the answer is that glycolysis takes place in the cytoplasm, while Krebs cycle takes place in the matrix of the mitochondria, while ETS is now taking place in the inner membrane of the mitochondria. Simple. So, sir, you are talking about we have to increase the proton concentration in the intermembrane space. So, to create a proton gradient, we have to also decrease proton concentration where? In the matrix we have to decrease. So what will be the electron passage? Electron passage will be from matrix to IMS, that is intermembrane space. This will be the electron transfer from matrix to IMS. So note down this. This is the basic idea of ETS. In photosynthesis we have studied ETS, and this is somewhat similar. Main necessity, create proton gradient. For that we are doing this all steps. Now I am rubbing the board. Next, to create a base, I will start with basic. For this, we require 5 complexes. We need 5 complexes. Let's write name of each complex. One by one we will write. So complex 1. Complex 1. Complex 1. Complex 1 is NADH dehydrogenase complex. So what is included in it? F F F yeah, wait, FMN and yes, FMN and FES. FMN is flavin monosulfate, means flavin FNM of FMN. So full form is not required. Full form is not required. Just remember FMN and FES are present in complex 2. In complex 2. Complex 2. What is the name of the complex? Is succinate dehydrogenase. So here we require FAD and FES. Here we require FAD and FES. Complex 3. Complex 3 here is cytochrome BC1. It consists of cytochrome B. We will write cyto B. In short, for cytochrome I am writing cyto B, FES and cyto C1, cytochrome C1. Fourth complex 4. Complex 4, we need cytochrome AA3. Cytochrome AA3. So requirements, what are the requirements? Cytochrome A we will require, and cytochrome A3 we will require. Simple, by the name. Now complex 5. Complex 5 is ATP synthetase. ATP synthetase, which consists F0 F1 particles or the oxysomes, which we have studied in cell chapter. So note down the basic name of all 5 complexes. These complexes are present in the inner membrane of mitochondria. So note down this. Then we will start the main reactions and all. ETS, we will start our main ETS. So note down this. Okay. as you know, we got four reduced coenzymes, that is 3NADH and 1FADH2. So we will do oxidation. We will do oxidation of NADH and FADH2. First we will talk about oxidation of NADH. Heading, let's draw. This is the inner membrane. This is the matrix, and this is the intermembrane space, that is IMS. So oxidation of NADH we are doing, means in matrix we have NADH, we have NAD, H plus we have, which we got. So here is the electron transfer. Electron will move, then it will be taken by FNM, then it will be taken by FES. So this forms which complex? Complex 1, right? So here 2 electrons are transferred, 2 electrons. Then these 2 electrons are transferred to a mobile carrier, electron carrier, that is ubiquinone, which gets converted into UQH2, which gets converted into UQH2, and it takes 2 H plus ion, H plus protons from the matrix, 2 H plus protons from the matrix, and gives it into the IMS. After this, these electrons are given to cytochrome B, then cytochrome B to FES, then from FES to cytochrome C1. This forms complex 3. This forms complex 3. First we are doing oxidation of NADH, because in this process we have to do oxidation of NADH, means we have to recycle NADH. That is the main purpose. Okay, here we are there. So cytochrome C1 gives again to a mobile electron carrier, that is cytochrome C. Then the fourth complex, it gives to cytochrome A, cytochrome gives it to cytochrome A3. Then the electron is given to half O2 accepts it, half O2 is accepting the electron and taking plus 2H plus, forming water here, oxygen is reduced to water, and this water is called metabolic water. So oxygen, we can see here, is the ultimate acceptor of electrons. So here 1, 3 and 4th complex are playing a role. So we can remember it by 1, 3 and 4, simply 1, 3, 4 complex are playing a role. Now this you have to remember it. This complex gives 2H plus. This complex has an ability to give 4H plus, and this gives 2H plus. Now you can total it. Total will come 10H plus. Means you can see the concentration here is increased. the concentration here is increased and here it is decreased. Thereby you can see the flow of H plus or electrons. Proton gradient is created. This way oxidation of NADH is done. You have to remember this. Note this, then we will do oxidation of FADH2, which is the second process of this cycle. Okay, I am rubbing it now. Oxidation of FAD. Again we will draw a box. Here is the matrix. Here is the inner membrane. Here is the intermembrane space. So here is this FADH2. So now here the electrons will be given to which FAD. FAD is getting the electrons and this is giving to FES. It is giving to FES, means this complex is succinate dehydrogenase complex, that is complex 2. Then this is giving to ubiquinol. And ultimately ubiquinol is giving to cytochrome B, then to FES, then to cytochrome C1. Cytochrome C1 is giving to, this is the third complex you all know. Cytochrome C1 is now giving to a mobile electron carrier, cytochrome C. Cytochrome C is giving to the fourth complex, that is, which consists cytochrome A and cytochrome A3. Again these electrons will be picked up by oxygen, plus it will take 2 H plus and will give H2O, which is metabolic water. Here 2, 3 and 4th are playing a major role. Here 2, 3, 4 complexes are playing a major role. So I can write it 2, 3, 4. Remember, oxidation of FADH2. So this does no pumping. No pumping is done by this complex. No pumping. As usual, this will pump 4H plus. This is pumping 2H plus. So what is the net here? 6H plus. Again a proton gradient is created. More concentration in the intermembrane space than the matrix. So this is oxidation of FADH2, which we have done. Quite simple process. Just here there is absence of complex 1, and in oxidation of FADH there is absence of second complex, and here there is absence of the first complex. Simple as that. Now in photosynthesis we studied some term which was chemoosmotic hypothesis in NCRT. So the fifth complex, here I can draw F0 particle, and here I will draw F1 particle. So this is the inner membrane, this is the matrix, and this is IMS, that is intermembrane space. So you can see proton gradient was here. The transfer of electron was in this way. Proton gradient, here there were more proton and here there were less proton. So now we have to break the proton gradient. That is, will transfer proton here. You're transferring proton here, and due to the conformatory change in the F1 particle, we are producing ATP. ATP means by breaking the proton gradient created, we are producing ATP. And in the previous video I told you that one NADH produces 3 ATP molecules and one FADH2 produces 2 NAD molecules. So this is ETS. We got the ATP molecule here. This whole process divided into 3 steps. First, oxidation of NADH, second, oxidation of FADH2, and third, breaking of the proton gradient. And here the complex 5 is playing a role. Complex 5 is playing a role doing chemoosmosis and producing ATP. So this is ETS. Now let's come into the next topic, that is respiratory balanced sheet. We will just total the account of ATP. That's it. It's a simple sheet, balance sheet we will write. Provide you a balanced sheet. Balanced sheet. First was glycolysis. In glycolysis, remember the products: 2 ATP, very good, 2 ATP, 2 NADH. Now, if one molecule of NADH produces 3 ATP, then 2 molecules will produce 6 ATP. Total in glycolysis we got 8 ATP. And double dashing this. Second link reaction. In link reaction, what was the product? 2 NADH. That means 6 ATP. 2 into 3, 6 ATP. Now third, in Krebs cycle, remember the products wisely. For one glucose molecule, we have to perform 2 Krebs cycles. So 3 NADH were formed in first Krebs cycle, so into 2 I will do, and 6 NAD will give 18 ATPs into 3 karke, 18 ATPs. Two ATP or GTP were formed by SLP, substrate level phosphorylation. 2 ATP. 1 FADH2 in the first cycle, into 2 cycles are there, because, and again into 2, because 1 FADH2 produces 2 ATP, so 4 ATP. Now adding this, we will get 24 ATP. Now add the total, 14 plus 24, 38 ATP are produced. So this is the balance sheet. First glycolysis, link reaction, Krebs cycle. This is the balance sheet. Now, in the next video, we are left with, I will cover amphibolic pathway and respiratory quotient. These are very small topics. I will need just 10 minutes to cover these topic, and then we are finished with this chapter. So I will tell you what we studied today. We studied important ETS, that is electron transport system, and the balance sheet, that is respiratory balance sheet. And that's it for today's lecture. Thank you for watching. So today's I will keep a goal of 100 views for today's video in one day. So share with your friends and with your class 11th students, 11th or 12th, you can share the video. So thank you.